All right, kiddos, let's talk about Objective 1C. And so Objective 1C is going to cover all things cells. So we're going to have our cell structure and function, and we'll talk about the different types of cells and the cell organelles. So let's start off first of all with reviewing what a cell is. And a cell is simply the basic unit of life, or the basic unit of structure and function for all living organisms. Now, there are two main categories or classes of cells. And so the first one is going to be the most ancient cell, which is believed to have been the first cells, which is going to be your prokaryotic cells, which are found in prokaryotes. Now, the thing about a prokaryotic cell is pro means before, karyote roughly translates into nucleus. It's actually like another kernel, but it roughly translates into nucleus. So a prokaryote is a cell that was before the nucleus. So there's not going to be any nucleus in a prokaryotic cell. Prokaryotic cells are also simple cells. They only have the necessary organelles they need to survive. Now, I keep saying organelles, but let's make sure we understand what an organelle is. So an organelle is the thing inside of a cell that does the jobs of the cell. So it does the work that maintains homeostasis inside of a cell. So these are things that's going to be inside of the cell. Okay. Now, there's going to be no nucleus inside of a prokaryotic cell. There's also not going to be any membrane-bound organelles. So remember when we talked about carbs, lipids, proteins, and nucleic acids, how there's a lipid called a phospholipid, and so that phospholipid has a hydrophilic head, and then that hydrophilic head also has the glycerol and the two fatty acid tails attached to it, and those fatty acid tails are hydrophobic. So in an aqueous environment, they tend to arrange in a bilayer because the heads like water and the tails don't. So they arrange in a bilayer so that the hydrophilic heads are in contact or attracted to the water that is found in the cytoplasm and also in the extracellular fluid, whereas the tails, which are hydrophobic, are protected from that aqueous environment. So the phospholipid actually forms these membranes, okay? The main one being the cell membrane. But there's also organelles found inside of eukaryotic cells that are surrounded by membranes as well, the nucleus being one of those organelles. So prokaryotic cells are simple cells. The only membrane associated with a prokaryotic cell is going to be the cell membrane because, of course, you have to have a cell membrane to have a cell. Now, the prokaryotes are found in two domains, archaea and bacteria, and each domain has one kingdom. Archaea gives you your archaea bacteria or your ancient bacteria, whereas the bacteria domain has the kingdom of eubacteria, which are going to be your true bacteria that we are most familiar with. And so it's fair to say that prokaryotes are actually bacteria, right? Because I have the ancient bacteria, then I have my true bacteria, which all fall under the prokaryote type of cell. So bacteria are prokaryotic and prokaryotes are bacteria. Now, the next class of cells is called the eukaryote. The prefix "eu" means true, so they're going to have a true nucleus. These are cells that have a true nucleus. So what is a nucleus? A nucleus is essentially a membrane that separates the genetic material. This is my nucleus here. That separates the genetic material from the rest of the cell. Okay. So if this is my cell, this is the cytoplasm out here, then a nucleus is going to be a double membrane that's going to be used to separate the genetic material or the DNA from the cytoplasm. So that's what a nucleus is. Now, inside of the nucleus, there is a condensed area of DNA called a nucleolus. And so that's extremely important because the nucleolus is going to make the subunits of an organelle called a ribosome. And we know that ribosomes make proteins. And why is that important? Well, DNA is transcribed to RNA, RNA is translated to proteins, and proteins are expressed as your traits, except for when they're not. All right, but anyway, so my second class is called the eukaryotes. And so this is going to fall under the domain eukarya, and all other living things fall under this domain. So I have 4 kingdoms. Protista, this is going to be your protists, things like Eugenia, Paramecium, and amoebas are going to fall under protista. Protists are typically going to be unicellular, and they have different modes of getting their nutrients and even their structures. In fact, the only thing the members of this kingdom tend to have in common is that they are made up of eukaryotic cells. I often call this the junk drawer. I said everything that didn't fit somewhere else but had a nucleus was put into this kingdom. Next kingdom you have is the kingdom of fungi. Now this is going to be your fungus. These are going to be 100% heterotrophs. These guys are going to have cell walls made of chitin. And so they can be unicellular or multicellular. Think unicellular like yeast or multicellular like mushrooms. So this is going to be your fungi. Next we have the kingdom of plantae. These are going to be your plants. Plants are photoautotrophs. So they are able to transform sunlight into that light energy or radiant energy into chemical energy using a special organelle called chloroplast. These are going to be multicellular, and again, they are producers because they are autotrophic. And then the last kingdom is Animalia. This is going to be your animals. These are going to be heterotrophs, and they are, again, multicellular, okay? So, everything else other than bacteria falls under the domain of eukarya. So this is going to be, they're going to have eukaryotic cells, meaning they are composed of one or more cells that contain a nucleus. Now, if we look at the actual structure of prokaryotes versus eukaryotes, my prokaryotic cells, again, are very simple, okay? There's going to be a cell membrane. They're going to have a single circular chromosome. So there's going to be DNA. The DNA is going to be in the cytoplasm, and there's only going to be one chromosome, and it's going to be circular. They have a single circular chromosome. They also have additional packets of DNA called plasmids, and these plasmids are going to help them to actually survive adverse conditions. We use plasmids also with biomedical engineering. For instance, you can take a plasmid and insert a human gene into it and get it to be expressed into the protein. That's actually how they make things like insulin. There's going to be ribosomes. Ribosomes make proteins. Proteins are expressed as traits, so all cells need ribosomes. Ribosomes also are composed of ribosomal RNA, so it does not violate the rule of no membrane-bound organelles because a ribosome is not a membrane-enclosed organelle at all. And then the prokaryotes are also going to have a cell wall. Now, a cell wall is going to be external to the cell membrane, and this is used for external structure and support. Some prokaryotes will have a tail-like structure. This is called a flagella. Flagella is used for movement. They may also have these hair-like structures on them, and these hair-like structures are called cilia. Cilia can be used for movement or anchorage as well. Now, notice that both the prokaryotic cell and the eukaryotic cell have DNA, but the location is different. The other thing that's different is going to be the structure. So in a prokaryote, single circular chromosome in the cytoplasm, but in a eukaryote, there's going to be multiple linear chromosomes located in the nucleus, and the number of chromosomes varies between species, between kingdom and species, okay? So different organisms have different number of chromosomes, but they are going to be linear, and they are going to be located inside of a nucleus. When it comes to the cell wall, you can find cell walls in every kingdom except for the animal kingdom. Animal cells are the only types of cells that lack cell walls. The cell membrane, of course, is found in every type of cell. Cell membrane's job is to control what enters and exits the cell, so it controls cellular transport, which helps to regulate homeostasis. It maintains a stable internal environment, separates the inside contents of the cell from the external contents of the environment. So all cells will have a cell membrane. And again, all cells will have ribosomes because ribosomes make protein. DNA is transcribed to RNA in the nucleus. That RNA is then translated to a protein at the ribosomes, okay? In prokaryotes, the ribosomes are going to be in the cytoplasm. In eukaryotes, you're going to have ribosomes in the cytoplasm and attached to the rough ER. for the actual cell, and then if the cells that have chloroplasts will have its own ribosomes, and mitochondria also has its own ribosomes, okay? So you're gonna find ribosomes in more than one location inside of a eukaryotic cell, or they're only in the cytoplasm of prokaryotic cells. Now, so the things that you're gonna find in all types of cells, okay? You're gonna have DNA, you're gonna have cell membrane, you're gonna have ribosomes, and the other thing that you're gonna have in all cells is going to be the cytoplasm. Now, cytoplasm is a jelly-like fluid that is going to be found in all types of cells, and that cytoplasm job is to hold the organelles in place, number one, but it's also going to be the site of many of the cell's chemical reactions, okay? So there's a bunch of enzymes and things that can be found inside of the cytoplasm. And remember, enzymes are just catalytic proteins. What were they made by? Ribosomes. All right, so those are the things that's going to be found in any type of cell. Now let's talk about some organelles that will not be found inside of your prokaryotic cells. And these are indeed going to be your membrane enclosed organelles. Now, your membrane enclosed organelles, first and foremost, includes the nucleus. And we already talked about the fact that the nucleus is the location of DNA. Okay, so this is where I'm going to find my DNA in a eukaryotic cell. So it's a eukaryote only. And because DNA controls the cell's activities, that's what we say the nucleus does. We say that it is the control center of the cell because that is where I'm going to find the DNA, which is going to be transcribed to RNA and then translated to proteins, which are expressed as your traits. Now, the next organelle is the mitochondria. Mitochondria is a double membrane enclosed organelle. Its origin is said to have come about is explained through the endosymbiotic theory and saying that a cell engulfed another cell and developed a symbiotic relationship. The job of the mitochondria is that it generates most of the cell's ATP. Now, the mitochondria only works in aerobic respiration. So during aerobic respiration, the mitochondria can produce anywhere from 34 to 36 ATP total. 2 from the Krebs cycle, and you can get 32 to 34 more from the oxidative phosphorylation, which includes electron transport chain and chemiosmosis. Now, this mitochondria is also going to be found in eukaryotes only, okay? The next thing you have is going to be your rough ER. The function of ER in general is to transport things. The rough ER has ribosomes attached. The ribosomes will attach to it. So the ribosomes is what makes it rough. And the proteins that are going to be secreted into this ER are going to be transported to the Golgi. Okay? So that's the job of rough ER. Proteins made on the rough ER are transported to the Golgi apparatus. Again, because it is membrane enclosed, this is going to be eukaryotes only. In fact, all of these membrane enclosed organelles are eukaryotes only. Your smooth ER. Smooth ER has 2 jobs. One, it can synthesize lipids, and it also detoxifies, okay? Detoxifies chemicals. So the lipids that are going to be synthesized in smooth ER are also going to be transported to the Golgi. So the ER works closely with the Golgi. Now, another thing about the ER is the ER is attached. It's an extension of the nuclear membrane. So your ER is going to be right there by the nucleus because it is an extension of the nuclear membrane. Our next one is the Golgi apparatus or Golgi bodies or just Golgi. And this one is used for sorting, modifying, packaging, and shipping any of those substances that are sent to the Golgi. So proteins, lipids, it does some things with carbohydrates as well. So the Golgi's job is to sort, modify, package, and ship. And most of the things that are at the Golgi are going to actually exit the cell. But it also does things like the Golgi is going to be the thing that's going to generate these lysosomes, because lysosomes contain digestive enzymes, okay? And these digestive enzymes can break down a cell. So the enzymes that are made, the digestive enzymes are made at the rough ER and transported to the Golgi where they're packaged inside of lipids as a lysosome. So it's like a storage container for digestive enzymes. And since I'm already here, I'll talk about it. The lysosome's job, they keep the cell clean. So think like Lysol, right? But they're only found in animal cells, okay? So even though they are found in eukaryotes, they're specific to the animal cell. Now, let's go back up to the vacuole because I kind of skipped it. Vacuoles are also storage containers. They can store water, food particles, waste, enzymes, salts. They can store different things. And what they store depends on the type of organelle they're found in. Type of cell they're found in, sorry. So if you have a large central vacuole, this is going to be found in plant cells only, right? It's going to be very large. It's actually the biggest organelle inside of a plant cell. You can find small vacuoles in other cells. They're usually food vacuoles that will then fuse with the lysosome to break down the food particles. Or you can have a contractile vacuole, which actually is used to remove water from a single cell, a unicellular organism like protists, okay? They may have a contractile vacuole that actually contracts to exert water from that cell. Vesicles are membranous organelles that are used to actually package things in and ship things in. So if something needs to get from the rough ER to the Golgi, or from the Golgi to the cell membrane, they usually butt off in a vesicle. So it's kind of like the Ubers of the cell, right? Different particles are shipped around inside of vesicles. And then another one, I put a star beside it because it's called a pseudopod, and the pseudopod is a fake foot. We see these in a class of protists where the cell membrane actually extends and then pulls back, and so it is used for movement. And so it's a fake, fake pod foot. So it's like a walk, so it helps the cell move like a foot, but it's a fake foot. So because it's an extension of the cell membrane is, yes, but it's the actual cell itself that is doing that. So now, another one, and I don't know how to put it up here. So I'm going to erase pseudopod from right here since I already talked about it. And let's talk about this other membrane-bound organelle, which is the chloroplast. Okay, now the chloroplast's job is to convert light energy into chemical energy. The chloroplast is a double membranous organelle with a third membrane system of thylakoids that contain a pigment called chlorophyll. And that pigment is able to capture light energy, and then that light energy through a process of photosynthesis is converted into chemical energy and stored in the bonds of carbohydrates. Now, this is done by photoautotrophs, so chloroplast is going to be found in your plant cells, okay? Chloroplast is going to be found in plant cells. So, plant cells will have chloroplast, animal cells will not. So, let's take a quick comparison between the different types of cells, okay? We're going to look at fungus, plant, animal, and prokaryotes, or bacteria. Now, first thing, prokaryotes will have a cell wall, will have a cell membrane, they will have DNA, but it's going to be a single circular chromosome. They will have ribosomes. They do not have a nucleus. The DNA is in the cytoplasm. They are simple cells. The biggest prokaryote is smaller than the smallest eukaryote, okay? So this is going to be my prokaryotic cells. Now, if I come over to my eukaryotic cells, they're all going to have a nucleus, a cell membrane. They're all going to have ribosomes and cytoplasm, which is also going to be found in prokaryotes. But every type except for animal cells will have a cell wall. Animal cells do not have a cell wall for structure and support. Instead, they use an internal structure called a cytoskeleton, or a cell skeleton, okay? So you're going to find that in your animal eukaryotic cells. Your animal cells are eukaryotic. In addition to that, they're going to have membrane-enclosed organelles like the ER, rough and smooth, and the Golgi apparatus. Different versions of a vacuole, although plant cells will be the only one that would have a very large central vacuole. In fact, it's the largest organelle inside of a plant cell. They're going to have mitochondria. Mitochondria generates ATP through aerobic respiration. You're gonna have in animal cells, you're gonna have a centrosome. Okay, these are animal cell only. Centrosomes are used for aiding in the cell cycle. They create the spindle apparatus that is going to create spindle fibers that will attach to chromosomes and move them around during the cell cycle. You're also going to have chloroplasts found in your plant cells, and chloroplast does photosynthesis. All right, and now when we talk about the centrosome, flagella, cilia, and cytoskeleton, these things are not membrane bound. They're made of microtubules or microfilaments or intermediate filaments. But the centrosome come in pairs found in animal cells. Flagella and cilia are both used for movement. A pseudopod is used for movement as well. And the cytoskeleton provides internal structure and support for these animal cells because they do not have a cell wall. The cell wall provides external structure and support for your plant cells and your fungal cells. Now, when it comes to plant cells and fungus cells, the way you can tell the difference, plant cells will have chloroplast and a large central vacuole. They both have cell walls, but the cell walls are made of different things. Inside of the fungal cell, the cell wall is made of chitin, and plant cell walls are made of cellulose. All right, guys, and it was a lot, but that was the difference between your types of cells and your cell organelles. Toodles!